Hello, thank you for joining us. My name is Sven Bokland, and I'm Director of Scientific Affairs at Bionano Genomics. I'm excited to talk to you about our genome imaging solution for high-throughput detection of structural variation in cancer and genetic disease. But I'll focus on cancer today. So when we're talking about structural variation, we're talking about these large rearrangements in the genome that can occur really all throughout. And if you're a scientist who's keeping up with the high-tech methods in long-read sequencing and all that, it may be a surprise to you that the standard of structural variation detection in a clinical setting is still standard cytogenetics. And what happens there is really a 50-year-old method where cytogeneticists use a microscope to look at intact molecules of DNA and visualize the patterns on these molecules. And by looking at changes in these patterns, they detect structural variation. So when NGS came around, there was this idea that if we could simply sequence all the letters in the genome, we would be able to see these structural variations just as well. But that hasn't actually worked out that way at all, mostly because these NGS researchers didn't come with a microscope. They came with this, with a blender. And this may seem silly, but I'll challenge you to find, to tell me how many pieces of strawberry were in this blender and where they were after I turned this machine on. But that is exactly what you do when you run an NGS experiment. The first step is that you take your beautiful intact genome and you shred it into tiny little pieces. And then you try to glue these pieces back together algorithmically to get an idea of what that genome looks like. Now imagine what would happen if you could just look at this structure without shredding it first. Makes a lot more sense. And that is one of the issues, but one of the complications with next generation sequencing also is that the genome is just really highly repetitive. It's just about a third of the genome consisting of unique sequences and 2/3 consisting of repeats. And now, all but one of these repeats that are mentioned here on this slide, for each of them, each unit on their own is already longer than the standard read length of say, Illumina sequencing, longer than the 150 base pairs. So now imagine making sense of long tandem arrays of these repeats with only 100 or 150 base pairs at a time. It's just not possible. And now you can say, I don't care about these repeats, but a lot of the structural variants in the genome are actually flanked by these repeats because they're caused by the repeats recombining through something like non-allelic homologous recombination or incorrect repair. And so if you're blind to these repeats in the genome, you're often also blind by the structural variants that are flanked by them. And that's why right now the standard of care is still for a cytogeneticist to look at these intact DNA molecules with a microscope. And that's exactly what Bionano Genomics does to look at the genome, except for we're adding a nanochannel array. These are hundreds of thousands of nanochannels in parallel, where we can linearize these DNA molecules, massive megabase-sized molecules, and image patterns on these molecules that we place on there in an enzymatic reaction. So think about karyotyping, but with a 500,000 times with 1,000 times higher resolution, 500,000 of these labels in the entire genome. Our microscope is called Saphyr, and it automates the imaging of these DNA molecules. Now, Bionano produces the instrument, the Saphyr instrument, but we also have our chips that linearize the DNA. We have kits for DNA extraction and labeling, and I'll tell you in a second how that works. And then we have our own analysis tools as well through Bionano Access, our software. Now, I want to get to cancer and how our technology plays a role in cancer research or diagnostics, but let me start by telling you how it all works. The first step, if you want to image megabase-sized molecules, is to get to megabase-sized molecules. That's important because every standard DNA isolation method, whether it uses spin columns or whether it uses magnetic beads or whether you simply precipitate the DNA, that will just shear that DNA. It'll rip that DNA apart by the mechanical forces exerted on it. And that's why we developed a protocol that is very, very gentle. Here we lyse the DNA in solution, and we use a disk similar in material to, say, a QIAGEN column, but it has a 3D structure that allows it to hold on to micrograms of DNA that are attached to that disk. Now you simply wash it and elute it in a new tube. And there's no mechanical forces, there's no grinding, there's no spinning, and so the DNA that comes off is megabases in length. The protocol is fast. It just takes a few hours. We're automating that right now. It works on fresh or frozen blood. We don't need more than 650 microliters of fresh or frozen blood. We can work with cultured cells. 1 million to 1.5 million is ideal. Or we can work with biopsies, cancer biopsies, for example, for as low as 10 milligrams. Keep in mind that we cannot work with FFPE-treated samples because the DNA is simply too fragmented, too damaged after that. However, we have other stabilization liquids similar to formalin, where you could, when you take a biopsy, simply put the material in that solution for up to a week before you freeze it. Now, if you look at this here, you get an idea of what other DNA isolation kits look like. They give you, for long-read sequencing, it shows here 20 to 30 kb. Now, if you place that on that graph on the left here with Bionano, you see that that's all the way in the far left, because our molecules have an average length of about 300 kb and go all the way up to megabases, as you can see in that tail there. So the DNA that you isolate with our prep method is just extremely long, nothing compared to other methods that are out there. The consequence of this, of course, is that if you have DNA that's already isolated on a sample, it's probably not going to work. It will not be long enough. Once we have megabase-sized DNA, the next step is to label a specific 6 base pair sequence motif. We're doing that with an enzymatic method that we call direct label and stain. Here, the enzyme will recognize that 6 base pair sequence and attach a fluorophore directly to the DNA. There's no nicking, there's no breaking. It's a single enzymatic reaction. And we use a single 6 base pair sequence motif all throughout the genome. So there's no probes involved. There's no multiple combinations of colors and labels. It's just a single label all throughout the genome. It's a 6 base pair sequence, and it occurs every 6 kb or so. And then the result looks like what you see on the top left there, really a ball of wrapped up, very long DNA with labels attached. And then we linearize that DNA. We use electrophoresis to pull the DNA into nanochannels. And these nanochannels are so narrow that a single molecule that's in that channel is uniformly stretched. And because of that uniform stretch, because it cannot coil up, we get all the information that we need just by taking a picture. And that picture tells us the distance between these labels, and we can convert that into base pairs. The array looks like hundreds of thousands of these channels. And you can see in this image below that every vertical line that you see is a molecule that's sitting in one of these channels shown above. The blue signal is the backbone of the DNA molecule that is stained with an intercalating dye. And then in green, you see that label that we attach to the DNA all throughout the genome. This is what it really looks like when the DNA moves through these pillars. It unwinds and goes into the nanochannels and then linearizes that. Now, the instrument will cycle molecules into the channels, image them, pull new molecules and image them over and over again. And that allows us to run 6 human genomes per 24 hours, or 42 samples per week. Later this year, we will increase that through an upgrade to the instrument software to 9 human genomes per week. Sorry, per day, 9 human genomes per day, so more than 60 per week. Or you can also run these flow cells simply longer and collect more data on that. So each of the chips have 3 flow cells. Each flow cell can collect 400x of coverage of a sample. We say 48 hours. In reality, it's a lot faster than that. So what this means is that you can collect a whole lot of data on complex cancer genomes. And I'll show you later what you can do with that amount of data. Also keep in mind that the cost per sample is just about $500, including the chips and the labeling reaction. So even if you collect 400x, if you run the chip longer, there's no larger expense. Once we collect these images on all of these nanochannels over and over again, our software will extract the digital information of these molecules. And then we perform a de novo assembly of the genome, meaning that we will align every molecule that we imaged with every molecule that we imaged all throughout the genome. And we find overlap, build an overlap graph like you see here, and then out of that at the bottom, a consensus genome map. And that consensus map represents the average spacing of each of the labels that we see all throughout the genome. Our software does this de novo, meaning that it's not reference guided, and that means that it doesn't actually know whether it's assembling a human genome or a tomato plant or a cockroach. We just built the genome that we see. Once that genome is built, we can then compare these maps to each other or to a reference. In this case, if the reference is in green, we will align our maps de novo assembled to the reference and look for alignment and look for differences. So in this case here, you see a deletion relative to the reference where the spacing between these labels decreases. Since we know the reference genome, we know where that 6 base pair sequence motif is throughout the genome. So that's how we can convert the reference into maps like this and do these alignments. Of course, all the knowledge from the reference still comes along, meaning that we know where the genes are, where the regulatory elements are, etc. So we can detect each major type of structural variant that way. Let's start with deletions. On top here, you see the reference in green. Your sample is at the bottom in blue, and you see that the spacing between these two labels decreases. When that spacing decreases by 500 base pairs, we call that automatically genome-wide as a deletion. An insertion is similar. The spacing between two labels increases, and at 500 base pair or more, that is an insertion. We can look at repeat arrays, where you can see that there's a change in the number of repeats. Sometimes you have a label per repeat, like shown here. Sometimes it's not the case, and then we can simply measure the flanking labels. We can detect tandem duplications. You see that this pattern here on top aligns on the left, but also on the right. The yellow labels are labels that simply don't align. And we detect tandem duplications, direct or inverted, automatically at 30 kb and above. A translocation is detected like this. We build a genome map in blue that aligns for half with one chromosome and for half with another chromosome or a different part of the same chromosome. We detect these genome-wide automatically, and the same is true for inversions, where we simply look for a pattern that's flipped around. Genome-wide again at 30 kb. And inversions are really hard to detect with sequencing-based methods. We're very good at that. So here again, you see the minimum sizes for deletions and insertions as low as 500 base pairs, and that's because we're just looking at the spacing between these labels. And then for duplications, translocations, and inversions, we need at least 30 or 50 kb, and that is because we're actually looking at complex patterns and how these patterns are changed. Now, when you first analyze a genome and you open our browser, our Bionano Access, you will see a circus plot like this, where each of the chromosomes are shown in the concentric circle, and the different type of structural variants are present there as dots. And then the purple lines in the middle are translocations between each of these chromosomes that are shown. So if you just click on any of these events, then you zoom in straight to the map, where you can see the de novo map aligning two different chromosomes in case of this translocation. And you can even show the molecules that were imaged by our system that support that translocation call. But on top of that, all of these structural variation calls will be filtered with a control database. So you can just filter out only the somatic events or the rare events that are not present in a control population, and annotate it, and it can be exported as a standard VCF file. I want to walk you through one example step by step so you get an idea of how that technology really works. So we spoke with researchers at MD Anderson who have been working on a family where 4 children have the same rare, very rare 1 in a million type of occurrence cancer. And the family had been studied by MD Anderson for a decade with no results. Here we're looking at a small part of the genome where we image some molecules. And if you look at these molecules, you see some patterns on there. So we build a consensus genome map that represents the average spacing of these labels detected on all of these molecules. When we align that to HG19 or another reference genome of your choice, you see that there's a perfect alignment on the left. Something's going on the right half. We also assembled a wild type allele. Of course, is a diploid organism, and so we expect 2 alleles. And that one aligns perfectly with the reference. Now, what's going on here is that there's a 38 kb segment that is present in 6 tandem duplications. It is not present in controls that do not have this tumor. So this is a clear genetic predisposition. And what's interesting here is that this gene, the large gene on the left, is known to be mutated and is in the pathway that is involved in this cancer. So what's interesting is that we are so certain that that structural variant is really there because we have single molecules that span the entire 230 kb of that repeat. The fact that these molecules exist means that that structural variant is really there. This is not determined algorithmically by chopping up the whole genome and trying to calculate how many reads you're expecting or something like that. No, we see a single molecule, and that molecule has 6 times the 38 kb pattern on there. So structural variants are observed directly on single molecules and not deduced algorithmically. Recently, 2 studies came out that compared our structural variation calls against some other technologies. One here of the 1,000 Genomes project looked at 154 human samples from 26 different ethnic groups and compared them against compared to Bionano data against data generated by Illumina sequencing by the 1,000 Genome project. And even though the study was done years ago on our old instrument with an older labeling chemistry that was much less sensitive, still we detected 8.5 times more insertions and even more deletions as well than with whole genome sequencing. And specifically, insertions are almost impossible to find with NGS because the aligners simply don't know where to place that extra information. And then more recently, a study was published from the Human Genome SV Consortium as well, where they compared Bionano against NGS and PacBio sequencing as well. And there's a lot of data here, but if you look at this graph here, deletions on the left, insertions in the right, starting at 500 base pairs and then going to megabases in the middle, you see this large section of bright blue information. Those are structural variants that were only detected by Bionano. Those were missed by whole genome sequence. They were missed by PacBio as well. So if you're not using a technology like ours that can detect these large structural variants, you're missing out on so much information. Now, I talked about repeats. I'll go over this real quick, but there's an interesting disease called FSHD, which is a form of muscular dystrophy. And it's the only known disease in the human genome that is caused by contraction of a repeat. All the other repeat-associated disorders are expansion disorders. And in order to genotype this correctly, you need to distinguish between the pathogenic 4qa allele and the non-pathogenic 4qb. You need to be able to know exactly how many repeat units there are, because if the number drops below 10 or 8, then a transcription factor gets turned on by a change in chromatin structure and causes the disease. Now, on top of all of this, there's also an identical repeat on chromosome 10 that is not involved in the disease, and you need to distinguish between the two. It's really complicated. With Bionano, you just look at it and you see how many calls there are. So later this year, there's going to be most likely 3, but for sure 1 diagnostic test being offered by some of our customers for this disease, clinical test, and we're really excited about that. Now, most expansion repeat disorders don't have a label per repeat unit like we showed there. In that case, we just measure the flanking labels, and we can size that interval and size that expansion. This was done here with myotonic dystrophy, which is a repeat expansion disorder, just like ALS or Fragile X. And you can see here on the right that the expanded repeat has a much wider distribution because this repeat becomes unstable in the tissue. And so within a single muscle biopsy, you see a whole range of sizes. And then the last sort of genetic disease example that I want to show you before I move on to cancer is here a study done by Mass General Hospital from Harvard, where they had a number of patients with a severe congenital disorder that were mapped with Bionano. They had a custom microarray that called a 2.3 megabase pair duplication in this area. But when we ran that with Bionano, we found that our copy number tool, that just counts the number of molecules all throughout the genome and normalizes that, found that there was actually 2 separate increases here. But more importantly, our de novo assembly shown below there shows where these duplications actually are. You see that this largest duplication on the left aligns to the left and to the right. And then the second duplication on the right aligns pretty much straight down, but also an inverted duplication to the left. That overlaps with that first section, and so that large gene there at the breakpoint is probably interrupted. There's a whole lot of information that we provide that you miss when you only get an array call that tells you that there's more or less of something, but you have no idea where or how. that duplication happened. Let's talk about cancer. This is a very old paper, but it still really stands out. It's a few years old. It's not that old. From the Garvan Institute in Sydney, where Professor Hayes looked at prostate cancer and analyzed the prostate cancer sample and the blood from the same patient using Bionano technology, but also using whole genome sequence on their Illumina X10s. And if you've ever tried to call structural variants from NGS data, you know that there's a whole lot of algorithms that do different things that have pros and cons. Some are more sensitive, some have more false positives. And so what you do is you run a number of different algorithms and you look for overlap. And so that was what was done here compared with Bionano. And the paper shows a large number of variants like here. This is a busy slide, but I'll walk you through it. In the middle there, in orange or red, you see the Bionano map in red, and the Bionano map from the blood of this patient. And right below that in blue, the Bionano map from the tumor of the patient. And between that in orange, a 14 kb somatic deletion that was detected using our technology. There's some variants relative to the reference shown above and below. And according to the Genome browser, that overlaps with a self-alignment or duplication. And that is probably why this variant was missed entirely from the Illumina data, not detected by any algorithm. However, if you look at the read pairs shown below there in red, those are read pairs that validate that deletion call. But in the context of all the other reads, those were missed by the aligner. And so what they found is that using Bionano, they found 85 of these large insertions and deletions, most of which affect genes, of which only 9 were picked up by NGS. Yet almost all of them were validated by manual inspection of these NGS reads. And there were a number of oncogenic driver mutations never before seen in prostate cancer. So really great results right there. And Dr. Broach showed something similar. This is right now in bioarchives, this paper, but it's submitted for peer review. And he looked at 12 leukemia genomes as head of the Penn State Institute for Personalized Medicine. And what they're showing in this paper is a number of variants, again, missed by other methods, but found with Bionano. For example, in the case of this Non-Hodgkins lymphoma, where the germline cells were mapped by growing the T lymphocytes that are unaffected in this disease, use them as germline controls. They show just a deletion, probably not relevant, but in the actual non-Hodgkin's lymphoma cells, there's a 3 megabase inversion right next to it that cuts right through the PTEN gene, that breaks the PTEN gene in half. And this inversion was not seen by whole genome sequence, and it was not picked up by karyotype or any other cytogenetic method either. And PTEN is a known tumor suppressor, and there are dozens of trials going on for compounds that rescue PTEN mutants. So the fact that something like this that could really change the outcome of the treatment was missed by other methods, picked up by Bionano, gives you an idea of the importance of having a method that can detect these structural variants genome-wide in any cancer sample. And so the next figure actually builds on that, because on the left you see a list of known leukemia driver genes that they found were affected by multiple variants, multiple structural variants in these 12 leukemia genomes. No surprises there, really. But then if you look on the right, you see a long list of known cancer-associated genes that were not before associated with leukemia, that have recurring structural variants as well. So now these are all genes for which there may be applicable drugs that target them. There may be prognostic value in these genes, and they've just not been checked. So if you're in a clinical setting right now, if your leukemia genome is being analyzed, they will not find these variants, but they would with Bionano. And then on top of that, there were dozens more of genes that were not known to be cancer-associated in the first place, and yet recurring structural variants occur in these genes. So an enormous potential here for discovering of biomarkers, of new targets, or of genes with prognostic value, really. And then lastly, they found that there were a significant enrichment of known cancer genes nearby SVs picked up with Bionano. So those are probably regulatory elements that were taken out by these translocations, inversions, deletions, or duplications. And again, there's an enormous amount of information here that's really not been studied carefully yet. That could give you a whole new career path, if you like. So Dr. Broach's paper is in bioRxives. He also recorded a webinar, which is available from Genome Web, and there's a transcription of that as well. You can find both of those on our website. And speaking of an inversion earlier, I want to show you an unrelated example here of a sample that was mapped by Bionano in a colorectal carcinoma sample. And here you see this 10 megabase inversion at the telomeric end of chromosome 9. But interestingly, the Bionano map at the bottom here extends past the reference. And that is because there's a telomere-telomere fusion of 9p to 9p. This is something that you cannot detect with any other molecular method because these telomeric and subtelomeric repeats get in the way. In Bionano, our molecules are hundreds of kbs in length, so they anchor right through these repetitive areas into the rest of the chromosome. And so we can detect this telomeric fusion, which again is almost impossible with other methods. So I spoke about our copy number tool. I spoke about our de novo assembly tool. So with our copy number tool, and again, here we're just counting the number of molecules all throughout the genome and normalizing that to detect large changes, large deletions or gains. We can detect these copy number variants larger than 250 kb down to 10% allele fraction. And that means that if just a fraction of the cells in your tumor have copy number changes like this, our tools still can pick those up, similar to what array can do, really. But on top of that, we also built a new rare variant pipeline. And this pipeline allows you to detect low allele fraction structural variants with very high sensitivity. You can't really lead, you see right there that we have 90% sensitivity to detect each of these type of SVs, translocations, inversions, deletions, insertions, and duplications, down to 5% allele fraction. And again, 5% allele fraction means that if just 10% of the cells in a tumor have a heterozygous, say, translocation, we will still pick that up. And this is completely unbiased. There's no PCR amplification. There's no panel here. We're not just looking for events that we already know. Now we can show you any translocation with any translocation partner all throughout the genome at such low allele fraction, even as low as 1.5% if you combine 3 of the flow cells on our chip. And this very subtle slide here tells you how that compares. No one else can do this. No other technology comes even close to this type of performance, not just for $500 per sample. So I want to tell you about a few presentations that happened in the last few months at various conferences. These are all unpublished results. We have permission to show that to you, but you won't find it in a publication yet. So that's why I want to summarize them real quick. We have recordings of each of these talks at the bionanogenomics.com website under the resources in the video section. So have a look there if you want to see more of them, but I'll tell you the conclusions real quick. First, Dr. Levy presented at the Cancer Genomics Consortium conference about a consortium that he is running with multiple cytogenetics groups in the United States, including the Mayo Clinic, the PATH group, MD Anderson, Columbia, etc., University of Washington, where they are comparing Bionano against standard cytogenetics tools in cancer. And so the standard of care for leukemia is karyotype and then up to 10 or 12 FISH tests. Now, what they did here was run a number of genomes on Bionano and compare them against the standard cytogenetic test. And they show examples like this one here, where there's a large inversion actually on chromosome 16 in an AML sample. They compared the Bionano call against the FISH call. And as you see here in Bionano, we get exact breakpoints in the genes, something, of course, that you can never do with FISH. They compared our calls against microarray calls. So, for example, for this CDK66 gene deletion, you see on the left the Bionano map calling 219 kb, 218.7, and then with array, almost the exact same 217 kb deletion was called. So just spectacular overlap there. So of all the samples that were shown, there was one where there was absolutely no data that made any sense. It was a sample mix-up turnout. But other than that, there was a complete concordance between the Bionano results and the standard cytogenetics results, so karyotype, fish, and array. And this was done in a blinded fashion. This study is ongoing on a much larger sample size, but for now, those results are just spectacular. And then also at the Cancer Genomics Consortium, Rashmi Kanagal-Shamana from MD Anderson Cancer Center talked about MDS, myelodysplastic syndrome, a precursor of AML. And again, there she looked at a number of samples of a preliminary results of a larger study, and she found just incredible concordance. I'll show you just one example here of where we picked up a variant that was seen on a karyotype. The karyotype showed this translocation here between chromosome 1 and 12, but it couldn't see this complexity here, these multiple translocation events. And so when you zoom into that, you see that our software calls out that there's a section from chromosome 1 and 12, but also a section from chromosome 5 that was missed by standard cytogenetics. So in several of the cases that she presented, Bionano found extra information that went beyond what was known with standard clinical methods. I highly recommend that you look at these presentations on our website, and the same is true for this third study here by Alex Huygen from the Radboud University in Nijmegen in the Netherlands. They've always been a pioneer in bringing new methods into the clinic, and so they're doing a large validation study on 100 leukemia samples and also on 50 samples with constitutional cytogenetic abnormalities. And that study is far from complete. They have about 36 that they reported on. But in all of these samples, Bionano detected all known clinically reported variants. And so Dr. Huygen, in his conclusion, really calls for a cytogenetics revolution with this technology. We hope that he's right, of course. So to summarize how this works, how we look at cancer genomes, you see complex genomes like this one here visualized in a simple view, in a single view, where you see all the translocations, all the different deletion and inversions, duplication events. And there's an enormous wealth of information. So if you click on just one single of these translocations, I'm showing there with that little arrow, you're taken to a map that shows you the pieces of chromosome 7 and chromosome 10. Here there's an inverted part of chromosome 7 that was translocated to a part of chromosome 10, where there's a section followed by a 1.6 megabase deletion, then an inverted 210 kb section, then another 400 kb deletion before the rest of the chromosome resumes. So this very complex assembly here, but we have single molecules that span the entire array of this fragment that I just described on single molecules. So again, you cannot be wrong with this type of data. There's a software partner called Genox, which provides online annotation and classification of whole genome sequence. And we've partnered with them to make detection of structural variation much better. Because they are used to working with whole genome sequence, you can take your Illumina reads and simply upload them to their cloud, and they will call variants and give you a clinical report. Now, this doesn't work for structural variants because you just cannot call these SVs well from that data alone. And that's why in this little animation, you see that if you have read pairs trying to align in this region, the aligners do the best job they can to place them there. Now, when Bionano calls a structural variant in that region, in this case, say a 6 kb deletion, their new software can now use that knowledge to do a better job at aligning these reads. So they can push them apart like you see here and align them to that part of the genome and now get sometimes single base pair resolution, right, at these breakpoints. So at once you have a validation of the call, the Bionano call with NGS data, and you also get much better breakpoint precision. And so it's as if there's a new sequencing technology now that combines the sensitivity for structural variants that only a genome imaging or mapping solution like ours can provide, combined with single base pair accuracy of short read sequence. Finally, I want to point at a paper from one of our customers where they use their method to map long-range DNA methylation on single molecules. And of course, this can be interesting in cancer as well. This paper used a method to label unmethylated recognition sequences of an enzyme, green here, and then they used the red label to label standard Bionano structural variation detection. And so now you have the green methylation signal that overlays on the same molecule where you're also detecting structural variants. And it allows you to check methylation and SVs in a single assay on single molecules and then phase all of that about over hundreds of kbs. So now you can check that if a promoter is methylated here, that another promoter 200 kb apart is methylated as well or not. This type of data on single molecules, phasing methylation over hundreds of kb, that just doesn't exist right now. So this paper is a proof of principle paper, but we cannot wait to see what you would do with this type of power. So to summarize, I think in cancer research, there's just a wide range of applications of this long-range SV detection method. We can discover new fusion gene and help with targeted therapy development. We can help with biomarker discovery and find new genetic markers for disease progression or prognosis of therapy response. We can help culture cell lines and find out genome instability and repair mechanisms. And of course, our technology can be used to look for target effects in CRISPR and genetic engineering studies, etc. as well. We have results on all that. I would highly recommend that you visit our website. As I mentioned, there's videos under resources, but also click on the line all the way at the right, which is called Bionano U, and that is our blog. And you can see papers discussed. You can find the latest news. You can see video recordings from presentations from our customers at various conferences. And speaking of conferences, the day before ASHG in Houston, in a few days, there's a symposium, clinical research symposium, where we'll have speakers the entire day looking at genetic disease using Bionano technology. Some incredible presentations are coming. I look forward to those. And then at ASHG itself, we also have a workshop on Thursday. You don't want to miss that. There's be some big announcements there. So to summarize, our technology allows for unparalleled SV detection. We can find structural variants that are missed with other technologies all throughout the genome, even in heterogeneous cancer samples. Our answers are confident. We have extremely low false positive rates, and there's a high correlation with standard clinical methods. And we have a comprehensive solution where you can go from your blood sample to data to annotated structural variant calls in just 4 days or so. And with that, I like to finish and thank you for your time. And we're happy to take any questions.